Hello and welcome to Unit 2, and we're on Part 3 of Unit 2, Enzymes, and we're on Day 2 of Enzymes. So welcome to Enzymes, Day 2. Let's go ahead and begin talking about enzymes. And today's aim is what factors influence enzyme activity. So we're starting with two questions that should be a review from Enzymes Day 1. What is an enzyme and what is a substrate? So enzymes are proteins that speed up the rate of reactions that would otherwise happen more slowly, also known as organic catalysts. And we remember that enzymes are organic catalysts because they are proteins, and proteins are one of the four classes of biomolecules. So remember, all enzymes are proteins, but not all proteins are enzymes. Enzymes are a type of protein that controls the rate of chemical reactions. So what is a substrate? The substrate is the specific chemical that each enzyme acts upon. And remember that enzymes are specific to their purpose, specific to their substrate. And when they come together, they form the enzyme substrate complex, which is what we're about to talk about now. So let's talk a little bit more about enzymes and go on to Part A, notes. So the active site or binding site. The underlined parts are what you need to fill in on your paper. Enzymes are usually much bigger than their substrates. The protein chains fold to make a three-dimensional shape that has a specific pocket or site where the substrate can fit and interact. And remember that this is like a lock and key type relationship where the enzyme is able to fit into this precise shape like a lock and key. And this specificity of the enzyme is dependent upon its active site. So the way these things fit together is shape dependent. Let's see what we mean. Ooh. So we have the glucose substrate that is binding to the enzyme here. So you can see the enzyme is the larger piece, larger than the substrate. And as it comes into bond, the chemical reaction occurs. That chemical reaction occurs when the enzyme and the substrate form that enzyme substrate complex, and the resulting product is changed. However, the enzyme itself is not damaged or used up by the reaction and can continue to be used in the body. Enzymes are very specific and only work with certain substrates. The substrates move towards the active site, and then the product released, the enzyme is ready for more, is ready to be used again. Lock and key theory. Each enzyme is specific for one and only one substrate, one lock to one key. It is thought that in order for an enzyme to affect the rate of a reaction, the following events must take place. Enzyme substrate complex. So this is the enzyme, the larger piece. The substrate fits into that little hole, into that little active site because they match each other like a lock and key. And then the formed product at the end is the enzyme substrate complex. So in order for the enzyme to affect the rate of the chemical reaction, these four steps must occur. The enzyme forms a temporary association with the substrate or substrates. The enzyme and substrate form a close physical association between the molecules called an enzyme substrate complex. They bond. While the enzyme substrate complex is formed, the enzyme action takes place and the substrate is broken down into its smaller, simpler parts. An example could be a disaccharide becomes two monosaccharides, or a monosaccharide, two monosaccharides become a disaccharide. So the two things being separated apart, that is part of chemical digestion, which occurs in conjunction with enzymes. Enzymes help us digest our food. The opposite reaction would be the two monosaccharides forming together to make a disaccharide, and that's synthesis. After completion, so after the enzyme substrate complex has been created, the enzyme and the product separate, and the enzyme is then ready to react with another substrate. So after the enzyme substrate complex is created, either, let's say the starch molecule is broken down into two monosaccharides, or the two monosaccharides are combined to make a disaccharide, the substrate is changed, but the enzyme is left unchanged and ready to react again with more substrates. So you can see here, the substrate, sucrose, consists of a glucose and a fructose bonded together. So down here is the enzyme with its active site, and you can see the active site of that enzyme matches precisely to this molecule, the substrate of sucrose, which is glucose and fructose, two monosaccharides bonded together. So the enzyme and the substrate meet, the substrate binds to the enzyme, forming an enzyme substrate complex. Water is produced as a byproduct of this reaction. So the binding of the substrate and the enzyme places stress on the glucose-fructose bond, glucose-fructose bond, and the bond breaks. So as a result of that enzyme substrate bonding, at the active site, the bond between these two monosaccharides is broken. That's where we see that the substrate has been altered, has been changed by this reaction. The products are released, and the enzyme is free to bind to other substrates. So this part right here, this is the chemical reaction, where the substrate is changed from its original structure as a sucrose molecule, which includes these two monosaccharides bonded together. That chemical bond is broken by the addition of the enzyme. And that is part of the digestive process. Now that glucose and fructose are two monosaccharides freely flowing through the blood, they can enter the cell, and they're small enough to enter the cell and get into the mitochondria to be used as fuel for cellular respiration to allow us to create ATP, baby, and that's what it's all about. All right, so now let's talk about factors influencing the rate of enzyme action. And this first one, pH, we're about to do a lab on this upcoming week. Isn't that exciting? pH, it's interesting stuff, and it influences the rate of enzyme action. So pH, the optimum or best pH for most living things is close to 7, which is the same pH as my water. That's neutral. pH scale goes from basic on the high end to acidic on the low end. And it has to do with the concentration of hydrogen ions, but again, we're gonna do an entire lab on just this topic specifically. And the reason we're gonna do it right now is because pH is extremely important to the level of enzyme action. So high or low pH levels slow enzyme activity, and if the pH is in its optimum place, enzyme activity will be highest, and that's part of maintaining the homeostatic balance in an organism, is maintaining that optimum pH. A few enzymes such as gastric peptidase work best at a pH of about 2.0, and gastric peptidase, gastric, you might recognize this word, it's in your stomach. Peptidase breaks down proteins, peptides. You might remember that ACE means that it's an enzyme. Gastric peptidase breaks down proteins in the stomach. And 2.0, that's a very low, a very acidic pH. And the reason why that enzyme works best at that pH is because that enzyme is in our stomach acid. Pretty cool. Another factor that influences the rate of enzyme action is temperature. So temperature strongly influences enzyme activity. An optimum temperature for maximum enzyme activity is usually about 35 to 40 degrees Celsius, which you may recognize as about the human body temperature. Reactions proceed slowly below optimal temperatures or above optimal temperatures. And above 45 degrees Celsius, most enzymes are completely destroyed. So again, talking about maintaining that homeostatic balance in our body, maintaining homeostasis, this stable internal environment affects our metabolism. It affects the rate of chemical reactions occurring in our bodies. So we want to maintain this balanced, the same all the time or as stable as it can be temperature, and balanced the same all the time or as stable as it can be pH, to allow enzymes to work at their optimum rate in our bodies. As well, the third factor that influences the rate of enzyme action is the concentration of enzyme and substrate, meaning actually how much is present, right? Because the enzyme and the substrate act specifically with one another, if there's too much substrate, it can't all bind with the enzyme at the same time. When there is a fixed amount of enzyme and an excess of substrate molecules, the rate of the reaction will increase only to a point and then level off, right? It's because these locks and keys, they can only bind, the enzyme is unchanged, but it can only bind with one substrate at a time. So as a result, even if you introduce all of these complex carbs in our bodies, the enzymes that break down those complex carbs can only work as quickly as they can work. And as a result, there could be a slowed rate of reaction that doesn't match the increased concentration of substrate. So again, talking about this sort of happy medium, this is a very common theme throughout our study of biology. Living things work best in a fixed range. So this leveling off occurs because all of the enzyme is used up and the excess substrate has nothing to combine with. So it can still theoretically be digested or go through, and the reaction may still occur, but the rate is not going to be increased. The rate of the reaction is not going to be affected by the enzyme if it's all used up or it's all busy with other amount of substrate. If more enzyme is available than substrate, a similar reaction rate increase and leveling off will occur, because the increased amount of enzyme will react with all of the substrate at once, so there won't be multiple enzyme substrate bindings. Instead, all the substrate will be used up really quickly, and then again, a leveling off will occur because there's no more substrate. Then the excess enzyme will eventually run out of substrate molecules to react with and wait for more substrate to be introduced. All right, so that is it for your notes. You guys want to make sure that you go and complete your exit ticket, the Part B, check for understanding. And you also want to make sure that you are reading through all of the passages that are on your handout and annotating those passages as you went along and completed your notes with me today. All right, that's all I've got for you. So see you all on Zoom tomorrow. Have a super fantastic day. Don't forget to like and subscribe to my channel and comment on this video for more fun. All right, bye guys. Have a great day.